Clinical proteomics is an important new discipline, promising the discovery of biomarkers that will be useful for early diagnosis and prognosis of disease. Here, we report a new digital microfluidics, or DMF, method integrating several processing steps used in clinical proteomics, including protein extraction, resolubilization, reduction, alkylation, and enzymatic digestion. DMF uses discrete microdroplets of sample proteins on an array of electrodes and can be run at room temperature, allowing parallel automated analysis of samples. This methodology represents a significant advance over conventional methods and has the potential to be a useful new tool in clinical proteomics.
Hi, I'm Steve Shih from the laboratory of Professor Aaron Wheeler in the Department of Chemistry and Institute of Biomaterials and Biomedical Engineering at the University of Toronto.
I am Mais Jebrail from the Wheeler Lab at the University of Toronto.
And I'm Vivienne Luk, also from the Wheeler Lab at the University of Toronto.
I'm Aaron Wheeler. I'm fortunate to work with Steve, Mais, and Vivienne.
Today we'll show you a procedure for proteomic processing using digital microfluidics.
We use this procedure in our laboratory to study proteins in biological samples.
So let's get started.
Start the procedure in the fume hood by cleaning the glass substrates in piranha solution while wearing adequate protection. Piranha solution is composed of 3:1 concentrated sulfuric acid to 30% hydrogen peroxide, so care should be taken while working with it. Incubate the glass substrates in piranha for 10 minutes, and after cleaning, rinse them in deionized or DI water and dry the substrates with nitrogen gas.